Good evening, everyone. My name is Liu Sengping, and I'm an assistant professor in the Department of Ophthalmology at the National University of Singapore. On behalf of the Department of Ophthalmology and the Center of Innovation and Precision Eye Health at the National University of Singapore, I warmly welcome you all to the Innovation and Precision Eye Health Care webinar series. And this series aims to provide fresh insights into the latest scientific and technology advanced in the understanding and management of eye diseases. In this series, we are privileged to have leading local and international scientists and clinicians share and discuss perspective on the digitalization, decentralization, and the personalization of eye care in Singapore and globally. Before we begin, I would like to express my heartfelt gratitude to each of you to join us today. We are honored to have Associate Professor Zhu Lin with us for today's sessions. Prof Zhu is a leading researcher in the retinal biology, metabolisms, and the translational drug discovery at the Safe Sight Institute, University of Sydney. He serves as the head of the laboratory research unit and the head of the retinal drug discovery unit within the Macular Research Group. With a strong foundation in biochemistry and molecular biology, Dr. Zhu's work focuses on covering the cellular mechanisms driving the macular degeneration and related retinal disorders. Today, he will present a talk on building human macula on a chip platform to study disease mechanisms and test the new therapies. Prof. Zhu, the floor is yours.
Thank you, Zhenping. It's my pleasure to come here to give this talk. So may I start sharing my screen? Can everyone see this screen? Okay. Thank you, the panel, for inviting me to come here tonight to present the research program in our research groups. So our research groups is major focus on study human macula. That's why we call the macular research groups. So today I'm going to present the building a human macula on a chip platform to study disease mechanisms and test new therapies. So why we are so interested in the macula? Because most of the macular disease most of the macula have the disease when it start aging. So the human macula is a very small but very important part of the retina at the back of the eyes. So it will help us to see clear and fine visions. So this is the image of the macula. So give me a second. I just give I need a point, yes. Can you see the laser pen? Okay, so this is how the macula looks like in the human retina. So it comes from a fovea region and around 5 mm diameters, yellowish retina at back of the eyes. So most of the macula is very sensitive. When it start aging, you will get the age-related macular degenerations. So under normal condition, the macula is normal, while there's two forms of the age-related macular degeneration. Wet forms, which is the vascular changes, and the dry form is the atrophy degenerations. So people know the age-related macular degeneration is very the age-related macular degeneration, but people don't understand why it happened to the macular regions. So the main challenge here to study the macular-related disease is most of the laboratory animals, such as mice or rats, they don't have the true macula at all. So to overcome this problems, our lab tried to connect it with the New South Wales Eye Bank, and we start using the post-mortem donated eye tissue, human eye tissue, to study the macula. So we try to culture this kind of small piece of the eye tissues in the culture conditions and try to study how the macula develops the diseases. So until now, there's some progress on this model development. So we know this macula can react to the light just like a living retina, and it behaves same as the retina in the animals. But also there's some disadvantage of these platforms, which of course there's no working blood vessels because it's post-mortem tissues, and some connections between the key cells are missing. Why we start this research program to study macula, to establish the macula on a chip research background platforms? Because this model gives us a much more realistic view of what happens in the human eyes, much more than animals experiments alone can show. So what we can learn from this research platform is we can get insight into how cells change in macular diseases and what might help protect and repair them. So this is our research platform. This is second generations. Basically, it is formed comes from a trans wells, which in cultured in a 6 wells perfusion plate with a pump which perfuse our culture medium consistently for 24 hours. So this is this is how we set up in CO2 incubators. So this special lab setup that keeps donated macular tissues alive and healthy for more than 5 days. It's like a mini life support system for the macula. Besides, we study the normal human macula. There's also advantage that we can get diseased retina tissues, such as the macula from the diabetic retinopathies and age-related macular degeneration. So we can study the tissues, diseased tissue directly. Also, we also culture not only neural retina, but we also culture the choroid RPE complex. So we have the co-culture systems that we can put the layer of the choroid RPE underneath the transwells so that there is a very close contact with the new retina and the choroid RPE. So this is how it looks like under the microscope. So we can see the new retina, and this bottom, the grayish one, is RPE and the choroid complex. We also try to grow the RPE, primary RPE as a single layers. This is how it looks like, and we try to put the new retina on top of it to see how the interaction happen between the new retina and the RPE. So first, I want to show some quality control of our macula on chips, second generation of macula on chips. So this figure shows the fresh tissues, which is what we just isolated the human retina looks like. We use Hurst to stain the major nuclear layers of the retina, so should have the ganglion cell layer in the nuclear layer and outer nuclear layers. So we can see the clear structures. If we use the regular static culture systems, within 3 days, we can see the structure start disrupted. So the layers start disappearing and break down. While if we put this retina into our perfusion systems and culture for 3 days, we still can see a very intact 3-layer structure of the retina. If we do the statistic analysis and see the outer nuclear layers ratio to the whole retinas, we can see the macula on our chips actually has significantly increase of the structures integrities compared to the static controls. And we also use NAD plus NADH ratios as the parameters to showing the cellular activities, viabilities of the retina cells. So we can see the fresh controls, which has a normal, like standard NAD NADH ratios, has significant decrease if we use just a regular culture. While if we use our MoaC systems, we can recover this NAD ratio, which means they have quite good viabilities. So the conclusion for this quality controls is the macula on chip systems markedly preserve the retina natural architectures and maintain high cell viabilities, creating a stable and physiological relevant models for human macular research. So after we get these research platforms, we have another big research program is try to decoding the macular disease through multi-omics and AI. all these things is based on the human tissue-based disease modeling. So we can, the target will be the normal human tissues, macular tissues, diseased macular tissues, aged macular tissues, as well as we establish the stress modeling on macula chips. After we perform the experiment, we will do the multi-omics. Basically, we can do the proteomics, metabolomics, lipidomics from the same piece of the tissues so that we can get a very complicated view of the human macular tissues. Of course, we also establish different multi-dimensional biometrics to study. So we do the, instead of do the bulk omics, we also have established the single cell omics platforms. And right now we are establishing spatial omics to study the macula and different area of the human retina. Of course, with all this multi-omics approach, we will generate a large data set coming from the real human retinal tissues. While this large data set, the regular bioinformatics analysis cannot really handle more and more data. So we have a very close collaborations with the AI group, one AI group led by the Junbin Gao, Professor Junbin Gao. They try to use the graph neural network, try to put all these different layers of the omics data into one single graph. So by doing this, actually we can integrate proteomics, metabolomics, lipidomics data into one area, and so that we can identify potential treatment target or the cluster which cause the disease happening. And this cluster may include not only single layer of the molecules such as proteins, but also it will be a combination of the protein, metabolites, and lipids. So this is how we are doing the basic study based on this macula-on-chips research platforms. And here is one of the major progress. We try to use this I2AI program to fill the knowledge gap of the human macular metabolisms. This is a collaboration with the Associate Professor Jianhai Du from the West Virginia universities. And when we check the reference, we find actually people know very little about the difference, metabolic difference between the macular and the peripheral regions. That's how we get the human retina. We can see we separate the neural retina. This is the macular region and the mid-peripheral regions. We also get the macular RPE choroid and the peripheral RPE choroid. I have to say the RPE also shows significant difference compared to the peripheral. Macular RPE has significant difference compared to the peripheral RPE. So with the support from the Professor Jianhai Du, we use isotope labeling, try to trace how put the tracer into the culture media and to see how the human macula compared to the peripheral region to use different nutritions. We use C13 glucose. Also, we interested in the serum metabolism, plays very important roles in the 1-carbon metabolism, and nitrogen labeled glutamine to see how they are metabolizing nitrogens. We are interested in the metabolites taken by the new retina and RPE. Also, we are interested in how the released by the new retina and RPE. We try to understand how the different nutrition supply to each other between the RPE and the new retina. Of course, we also establish a different stress models such as light stress, which cause significant oxidative stress accumulating in the retina. And we also use like toxic lipids, deoxysphingolipid, which today I will introduce in details in the later. And sometimes we use high glucose to mimic the stress conditions as well. These systems also can use to test the different nutritions as the therapeutic cargoes, such as nicotinamide. We use serine treatment and pyruvate treatment. So this is how we are starting the experiment. So basically, this progress is we get the post-mortem eyes from the eye bank, and immediately we open the eyes. We will take the founder's photo of course, we want to make sure sometimes even there's no record of the retinal diseases, there's still some diseases happening. So we want to make sure this is all the normal retina, and we also take the backlight to see how the RPE and the choroid looks like. So the second one showing you a video is how we are dissecting and separate the neuroretina and the RPE choroid. So after we cut off this optic nerve, we take out the neuroretina into a separate dish, and then we start punch this neuroretina different areas like macular regions and the peripheral regions, and start culture in the on our macular on our chips platforms. So after we culturing, we can get the same piece of tissues, and we do the we call simplex extraction method. We can get the metabolites from the top layers. We can get a protein in the middle. This whitish is all the proteins, and the lipid at the bottom. So basically, we can get all these different omics and send to the mass spec and do the metabolics, metabolomics, proteomics, and lipidomics. So our pipeline use this post-mortem eyes on chips and multi-omics with AI. We try to use this to uncover the treatment target and identify the key cluster from the bulk, single, and spatial data. Our lab has another downstream research program we call nanovision program. This is more related to the translational discovery into the RNA lipid nanoparticle therapeutics. While in the past decades, the lab has identified so many different treatment targets, and we are trying to really translate into the clinic. We established platforms to from the design the RNA drugs, and with the different lipid nanoparticle formulations, try to selectively deliver the RNA drugs into different part of the retina, such as Müller cells. You can see this EGFP as markers, and the photoreceptors. And of course, we have the well-established the in vitro platforms and the in vivo platforms. But I want to especially mention here is because our macular on a chips research platform also plays very important role in the downstream drug validations. And we have already established several collaborations with pharmaceutical companies to test the drugs on using these research platforms. And our next 5 years goals is try to push these RNA lipid nanoparticle drugs into a real clinical trials. So this is the background of our research group's basic research. So today I will focus on talking about one research project which will show you the whole procedures, how we are using these platforms and to study a specific diseases. So today's topic will be the macular telangiectasia type 2. We call it MACTEL. MACTEL is a rare disease that only affects the center of the vision, which is the macula, and cause gradual vision loss. This MACTEL disease actually from 15 years ago, maybe around 15 years, 20 to 15 years ago, people know nothing about this disease. Gradually, with the support of the Lowy family, they established global collaborations, and people understand this disease, and gradually identify the potential treatment. So the following research data is coming from the research staff from our groups, Dr. Ting Zhang, Dr. Rita Zhen, and Dr. Qing Chen. They contribute all these major data for this part of the research. So some background, more background about MACTEL. So MACTEL is very specific disease. So it only happened a small oval-shaped area next to its center of the macula. So it never really passed through macular regions and confined in this area. Around 15 years ago, Professor Mark Gillis led teams to study the first donated human tissue with the MACTEL diseases, and they do the different stainings, and they surprisingly find actually in the macular regions when you the staining of the Müller cell marker has disappeared. That means this disease actually is starting with the losing of the Müller cell, which is a major glial cell supporting the whole retina. and the Müller cell marker disappear in the macular region, but still has a good standing in the peripheral regions. People don't know why this is happening. Around 10 years ago, Dr. Murray Gardner and his colleague and her colleagues started study identify a kind of toxic sphingolipid. We call it deoxysphingolipid. This sphingolipid could play a very important role in the pathogenesis of the MAC tail. So I will brief you some of the biochemistry background. So sphingolipid is a major important lipid supporting the neuroretina, and the urea is coming from the palmitoyl CoA will react with the help of the enzyme SPT will react with the serine, and for the sphingolin, the sphingolin will eventually become a sphingolipid to supply to the neuroretina. Under some disease conditions, such as a very high level of the alanine or the SPT enzyme has some mutations, the alanine will have the higher affinity compared to the serine, and this alanine will bind to the palmitoyl CoA, which form a deoxysphingolin. This deoxysphingolin will eventually become a deoxysphingolipid, and it's very hard to be metabolized, and it will cause a significant neurotoxicities accumulate in the retina. So that's why people believe the Dr. Gardner find actually there's a significant accumulation of the sphingolipid, deoxysphingolipid in the MacTail disease compared to the control. And also they have a very low serine levels. So they believe this is one of the cause of the pathogenesis of diseases. But there's still a major question remaining. If the toxic deoxysphingolipid is the cause of the disease, why this disease only limit to the macular region, but not to the peripheral regions? So that's why our lab started to study the specific metabolism in the human macula, try to solve these questions. So the first step we are doing this is because we know very little about glucose metabolism in the human macula. So we try to use the C13 glucose as a labeled C13 glucose as a tracer to see how the macula compared to the peripheral retina to metabolize the glucose. So this figure shows how we are doing this experiment. So basically, we feed the new retina with the C13 glucose, and then after 4 hours incubation, we will take the new retina, extract the metabolites from the retina, and we will do the mass spec to detect the different downstream metabolites. So the overall this PCA figures showing the overall. So this is around I think it's 12 to 15 different donors, macular region compared to peripheral regions. So we can see the downstream glucose metabolism metabolites has significant difference between the macular and the peripheral regions. And if we look into the downstream metabolites details, we find two major difference cause this difference between the macular and peripheral region. One is the lactate generated from the glucose. So we can see the macula actually generate significant higher amount of the lactate compared to the peripheral retina. And also, if we talk about the secretion, which is the retina takes the glucose and secrete out the lactate, also has a significant higher lactate secrete to the surrounding buffers, the medias in the macula compared to the peripheral regions. And if we use the lactate and the peripheral ratio to represent how the pyruvate converted into the lactate, we can see the lactate pyruvate ratio also significantly high in the macular region compared to the peripheral regions. So this actually, what does this mean? So this means the macula relies on a fast oxygen light energy pathway than the peripheral retina, probably to keep up the intensity amount of the central visions. Another significant difference between glucose metabolism between the macular and the peripheral region, actually we find is about the alanine, which is alanine has a very high, so macula generate a very high alanine total abundance from the glucose compared to the peripheral regions. And also they secrete more alanine into the median compared to the peripheral regions. Because the serine alanine ratio is the key to decide whether the chance of the deoxysphingolipid, the toxic deoxysphingolipid formations. We checked it. We checked the serine alanine ratios in the macular region and peripheral region, and we can see the macular region has quite lower serine alanine ratio compared to the peripheral region. So what does this mean? So this means the imbalance, more alanine and less serine makes the macula more prone to produce toxic deoxysphingolipid. So when we find this, it's just a hypothesis. So the next step, we're trying to test this hypothesis by directly profiling the lipid metabolism in the human macula. Similar like the stage 1 experiment, we take the macular regions and the peripheral region from the post-mortem retina, and we do the random mass spec and profile this sphingolipid profiling. So same as previously, we can see the significant sphingolipid profiling difference between the macular region, this is red, and the peripheral region, which is green. So what we found is normal human macula displays a distinctly different lipid metabolism profile compared to the peripheral retina. And then we further interested in whether it will accumulate more deoxysphingolipid compared to the peripheral retina. So we have to understand while we are using the post-mortem eyes, there's all different kind of conditions. There's a huge individual variance because we cannot control the gender, we cannot control the age, we cannot control the post-mortem times. So the best way to compare is to do the ratio which normalize to itself. So we use because the ceramide has different carbon chains. So we basically we use the deoxyceramide to compare the same carbon chains as a ceramide, which as an indicator to showing the level of the deoxyceramide accumulated in the macular regions. So when we check all the deoxyceramide to ceramide ratios, we can clearly see from different side branches of the carbon side chains, we can see the macular region has significant higher level of the deoxyceramide compared to the peripheral regions. Of course, there's another dihydroxy deoxyceramide, which is also very toxic. We did the same comparison. We can see the same trend. The macular regions has higher deoxyceramide compared to the peripheral regions. So this is come to the big picture is why people has low serine level get MacTail, because the macula naturally is easily to accumulate the deoxysphingolipid, while if the patient have the low serine level, this will make this accumulation more significant. But the toxic sphingolipid cause the retinal degeneration is never really tested in the human retina. So in the stage 3, our research programs, we are trying to test whether this sphingolipid really do harm to the human retina, both macular and peripheral regions. So we did a very complex experiment. We try to cut the macular regions because we have different groups. So we have to cut the macula into half, and also the relative different size of the peripheral piece as a control. So this SA means sphingolines, and this deoxysphingolines, which is one is the non-toxic control, this is toxic treatment. So we put into this sphingolines in the perfusion culture and put the deoxysphingolines into another diffusion cultures, and we put both left eye and right eye into different transwells. So after 3 days of the culturing, we collect different piece of tissue and do a series of the experiment, including the tunnel stainings, immunostainings, and RNA sequencing and Western blot. We got a lot of data. We try to test whether we expose healthy donated macular tissue to deoxysphingolipids will, in our MoaC system, will cause any harms to the new retina. Well, here I just show the most important data we got from this set of the experiment is this is the sphingolines controls. We did the tunnel assay. We can see some very mild tunnel tunnel staining in the ganglion cell layer, which is normal because every time we isolate the new retina, donor retina, we cut off the optic nerve, and it will cause some ganglion cells death. While for the deoxysphingolines treatment, we can see a significant increase of the tunnel staining in the inner nuclear layer. While inner nuclear layer actually is a major Müller cells nuclei is here. So we did the statistic analysis of the inner nuclear layers of the tunnels positive cells. We can see the significant increase from the 3 set of the donor test. And then we use the CRLBP, which is the Müller cell specific marker, to stain the co-stain with the tunnel assay. And we can see this enlarged image. We can see all this tunnel staining is being wrapped with the red, which means the tunnel staining happened to the Müller cells. So this solves the original. This is consistent with at the very beginning. I show you the clinical observations is when the McTail starting, the major cell disappearing in the macular region is the Müller cells. That is because this deoxysphingolipid treatment is extremely toxic to the Müller cells. After we understand the pathogenesis of why this macula have the special will or easily to develop the McTails, we are trying to use this knowledge to try to identify the potential treatment. Because we find the macular region has easily accumulated high level of the alanine, which is, we believe, the major contribution to this deoxysphingolipid accumulations. We are trying to see whether we can inhibit the alanine accumulation in the macular regions, while the de novo alanine synthesis is controlled by this ALT enzyme, which is a key enzyme control the glutamate react with the pyruvate will generate alpha Kg and alanine. Well, this enzyme has a well-established inhibitor, which is a BCLA, beta-chloro-L-alanine. It is alanine analog, and it has high affinity to this ALT. So when we treat with the BCLA, this analog will bind to the ALT enzyme and inhibit its further binding to the alanine. By doing this, it will inhibit the alanine biosynthesis. Before we start to the treatment, actually, we surprisingly find there's not a lot of study the ALT enzyme inside the human neuroretina. So we first did the staining of the ALT enzyme to see where this ALT enzyme is expressing in the neuroretina. And the ALT enzyme has actually two isoforms. One is called ALT1, which is a cytoplasmic enzyme. One is ALT2, mainly expressed in the mitochondria. And we use the different antibodies co-staining with the CRLBP, which is a Müller cell marker, and we can see the co-localization of this whitish co-localization suggests this ALT1 is mainly expressed in the Müller cell and some of the RPE, while for the CRLBP, for the ALT2, it also expressed mainly in the Müller cell and some of the inner segment of the photoreceptor, cone photoreceptors. So the ALT usually plays very important role, also involved in the help the cells resistant oxidative stress, all these things. So we are wondering whether inhibition ALT will do harm to the new retina or not. So we try to establish oxidative stress models using this macula on a chips research platform. So we use TBHP, which is inducer cause the oxidative stress accumulate in the new retina. And then we add the BCLA treatment, co-treatment. We want to see whether this inhibits the ALT will reduce the new retina's resistance to the oxidative stress. So this figure B showing how the TBHP models established. So we use NADP and NADPH ratios as the indicator of the oxidative stress accumulations. So when we add 800 micromolar of the TBHP treat with the new retina explant, we can see the significant increase of the oxidative stress. And if we use BCLA treatment on the retina together with under the oxidative stress, actually we can see some reverse effect, which means the inhibition of the ALT may not do harms to the retina. And we also use the LDH assay showing the toxicity cell toxicities, and when we treat with the TBHP, we can see some increase of the LDH assay, while the BCLA treatment can recover a little bit. So with this safety treatment, we then further establish to see what the BCLA can do in the alanine metabolisms. So as I said before, there's very little study about how the pyruvate alanine is metabolisms inside the new retina, because this is actually is a two-direction reaction. So pyruvate can metabolize to alanine, while on the other hand, alanine can change back to the pyruvate. So we designed an experiment which is using the labeled pyruvate or labeled alanine as a tracer. Then we feed the new retina and see the downstream metabolism. So in this figure AB, we're showing we feed the new retina with the labeled pyruvate, and then we detect the labeled alanine to see the alanine downstream alanine metabolisms. And we can see it here is after we inhibit the BCLA, the alanine significant decrease. That means the BCLA can efficiently inhibit the pyruvate metabolite to the alanine. On the other hand, we try to use the C13 labeled alanine to feed the new retina and detect the downstream pyruvate. While this actually is not that significant if we add the BCLA ALT into it, because macula shows no significant changes, while pyruvate has some changes but not statistically significant. It is quite easy to explain. So if we can see this percentage of the labeling, actually after 4 hours of treatment, if we add alanine, this very small amount of the pyruvate actually is coming from the alanine within 4 hours. While if we are talking about pyruvate as a tracer, and after 4 hours treatment, there's around 0.2 to 0.3% of the alanine is coming from pyruvate within 4 hours. So the conclusion is BCLA blocks the primary conversion of the pyruvate to alanine while having minimal impact of the reverse conversion from alanine to pyruvate. Because this ALT enzyme plays this alanine pyruvate reactions, it's actually play a very important role in both the nitrogen metabolism and carbon metabolisms. So we further want to see how these inhibitions will affect the nitrogen metabolism. So we feed the new retina with N15 labeled glutamines and to see how this labeled nitrogen goes. So theoretically, the nitrogen glutamine will come to glutamate, and with the help of the alanine, this nitrogen will transfer to the glutamate from glutamate to the alanine. There's some other transaminase reactions, such as go to the aspartate or go to the proline or go to the serine. So after we do the profiling, we can see if we add the 8 micromolars of the inhibitor of the BCLA to inhibit the alanine functions, we can see the significant increase of the glutamate, which means if we block this pathway, the glutamate will accumulate. And at the same time, yes, as we expected, the alanine levels have significantly reduced. On the other hand, the aspartate levels actually increased, which means aspartate is an alternative transaminase pathways to replace the alanine. So if alanine, this pathway being blocked, more glutamate has been transaminated to the aspartate. And also the serine has no significant changes. So the conclusion for this experiment is BCLA inhibits the transfer of the nitrogen from glutamate to alanine. In response, the cell partially compensates by increased nitrogen transfer from glutamate to aspartate. The next experiment is we trying this experiment with tracing the glucose metabolism, carbon metabolisms. So we, same as the previous ones, we feed the new retina with the C13 labeled glucose and profile the downstream glucose metabolites. So what we can see, actually the pyruvate don't have significant changes, while the serine, the periphery has a little bit response, but not that significant compared to the macular regions, while the alanine has significant down-regulated both in the macular region and the peripheral region, which means it can block the alanine biosynthesis, while aspartate for the carbon metabolism, there's no significant changes. So the conclusion for this set of experiment is BCLA inhibits the de novo synthesis of the alanine from glucose while having minimal impact on the production of other glucose metabolites. So back to the key things. We want to use this inhibition to treat the MACTEL. So we are really care is this, whether we can use this treatment to change the serine alanine levels. So from the glucose metabolism result, if we look at the alanine levels, it decreased. The alanine to pyruvate ratio also decreased. And the most important thing, this figure C showing the serine alanine level. If we do the treatment with the BCLA, they can significantly increase the serine alanine levels, which means the chance to form the toxic deoxysphingolipid will significantly reduced. So this result until now is changing the metabolism of the macula maybe help to protect against the disease such as the MACTEL. So we are still actually, this is the most updated result, and we are still actually checking the downstream whether there's a real changes of the oxysphingolipid accumulations after the BCL treatment. Well, today's take-home message until now is our model, which is the macula on a chips, help us to explore the metabolic difference between the human macula and the peripheral retina. So we identified actually when compared the periphery and the macula, periphery has more serine and less alanine, while the macula has less serine, more alanine. This imbalance actually will cause the deoxysphingolines accumulations, and it's very toxic to the neuroretina and also especially to the Müller cells and cause the Müller cell dysfunctions. While we are trying on the human macula, we test this BCLA as a potential drug treatment, try to reverse this balance and potentially can treat the MACTELs. So that's all for my today's presentations. And thank you to my teams and the supporters, especially thank you to our director, Professor Mark Gillis, give a very strong support to the whole research program. And thank you to the Lowy Medical Research Institute. We got continuous funding from the Lowy Medical Research Institute to continue this unique study. And Professor Meidong Zhu, coming from the New South Wales Eye Bank, provided strong support to provide the new retina, the post-mortem retina for our study. And Professor Jianhai Du from West Virginia University helped us to profile all this label tracing experiment. And Dr. Mari Gartner helped us to profile the lipid metabolisms inside the new retina. And the last is I would want to thank you to this generous donor and their family to donate their eyes for this research. Thank you very much.
Thank you, Prof. Zhu. It's a very nice talk and a very great work. You give us new systems of the human retina, human macula on a chip system, and also give us example how this can be used for the MACTEL's applications. Okay, so next we are going to do the Q&A sections. So it will lead by me and alongside the panelist teams, Assistant Professor Chen Hui-wen from the clinical department, and also the Assistant Professor Daniel Tay. And we are looking forward to engage all the audience to submit your questions in the Q&A sections, and maybe start from myself. So I have a few technical questions first. So the retina, how you can fix on the dish? What kind of glue and how do you do the fixations when you perfuse this?
Actually, I can understand your concern because basically the new retina, if we sometimes if we isolate it, it will curve immediately. While the trick here is we have to clear the vitreous efficiently. So vitreous is the biggest problem. So And also the size of the retina punch is also important. If it's too big, it's very easy to curve. While if you clean up the vitreous and the small punch like 5 millimeters, the biggest one we did like 6 millimeters diameters of the punch, the retina can like flat at the bottom of the transverse efficiently. And also, you can try the different membranes. So some of the membrane has affinity to the tissues. So if you put on top of it, it will attach to the membrane. So it will also will help the the flattening the new retina. While we didn't really use any glue or any other help extra help on flattening the new retina.
Okay, yes. My second question is, do you have any quality control factors to keep the this systems? So how do you define this is maximum can use for 5 days of the culture the retina still healthy? And what's the longest retina culture you can keep?
Actually, this is really good question. So, 5 days is a kind of average because we basically we use we publish a paper together with Jianhai Du as to study the like metabolic profile of the new retina, and we can see after we culture them for a while, the metabolite profile is still similar to the organoids and the mice retina. So that means they are still very healthy. While the how long it can culture, it really depends on the the quality of the postmortem delay of the new retina. So if the new retina in a very good qualities, I'm very confident it can culture much longer time compared to 5 days. Well, 5 days is a very safe time, but we won't see a significant change of the structure. We use Hertz staining, and the tunnel staining won't show a lot of apoptosis in the new internuclear layer and outer nuclear layer. While in the ganglion cell layers, due to the cut off the optic nerve, there's always apoptosis happening in that area. So that's how we are saying like 5 days is normal. Uh, I believe if we culture longer times, the new retina eventually will gradually degenerate. Well, yeah, so but it's really hard to say. Yeah, the quality control is it's a big problem because individual variance. While for our most of our data, when we compare, we always compare with the macular to the peripheral. So you will see a lot of ratio in our data. So that can help reduce the individual variance, and we always can see significant difference.
Okay, thank you, thank you. My third question is, have you considered using the different lights to stimulate the retina because it's a light sensitive tissues? Yeah.
Yes, that's a really good question. So, we try to use a like a blue light more to short wavelength more light to stress the retina, and we try to use very strong white light to stress retina. Well, the point here is because you know the vitreous actually can filter a lot of blue light. So we we still don't know what exactly wavelength or the strength of the light can reach the new retina, because when we do the explant culture, actually there's no protection from the vitreous. So we direct shining the light onto the new retina. So the we previously we set up this white light experiment is we can stress for 4 hours while there's a significant change of the transcriptional profile, but there's no cell death. That's how
Mm,
we establish this model.
okay, thank you. Yeah. So there's a question from the floor. So, with the age of the donate macula, does it matter to the results?
Yes, we are trying to, when we do a set of experiment, we try to control the age is around 50 to 70s, and we try to avoid to use too old donors or too young donors. While the age, on the other hand, is a huge advantage for using this macula on a chips research platform compared to others, because this is natural aged retina. So I believe based on our data accumulating, we are trying to also study the data across different batch of the experiment to see what the age will contribute to this disease. But at this stage, we don't have enough data to see the age difference. While we do select the donor's age to avoid too young age and too old age for the experiment.
Okay. Another question is so sorry, I have too many questions about the details. So when you culture the with the RP, have you seen any loss of the RP's pigments?
That honestly, this co-culture experiment, because I keep saying this is the second generation of our MoaC systems, and this co-culture is actually the third generation which we are developing at this stage. Um, Honestly, we didn't see the pigment disappearing because we still culture very just within 24 hours, so it's not a very long time. So we are trying to remodel these systems so that the choroid RP, because choroid RP also will curve, so we haven't really solved this problem yet. So we try to get it under more good conditions so that we can culture longer. So maybe we could see some changes for after 3 days. But at this stage, I have no answer to your questions.
Okay, thank you. Maybe I give the chance to the other panelists. Yeah,
Yeah, thank
please.
you, Prof. Ling Tzu. This is Daniel here. That's a great talk. I think most of the critical questions have been asked by Chen Ping, but I'll be asking more on the globe donation, because there's how much can you really control the time which you can receive the macula, the globe, because there's a lag time between retrieval,
Mhm.
and probably sometimes family members might want to do a wake or respect, and so I don't know at which stage the retrieval actually happens.
Okay, so basically, I think the iBank has two lines. So one is like direct donations, which it's more related to disease tissues. So a lot of ophthalmologists has a patient already signed the consent to donate their eyes for the study. So this will be very quick. So we once we got the MacTail eyes within 4 hours of post-mortem delay, because as soon as we know it, everything is already arranged. While for the
Four
normal,
hours.
4 hours,
Oh, wow, very
four,
fast.
four
Okay.
hours post-mortem delay. While for the normal one, which is irregular contribute to the eye bank for the cornea transplantations, it usually takes around 24 hours. Um, while I would say post-mortem delay actually is is a significant factors of the quality of the new retina, but it's not the only factors. So it's also depend on the donor's situations. So some donors, if they lie on the bed for very, very long times, the even very short post-mortem delay, the quality is not that good. While some fresh ones from the healthy donors, it even more than 24 hours, you still get very good quality. That's why we most focus on the post-mortem examinations. That's why we take the fundus photos, we collect all the tissues to see whether the quality is good or not. Well, honestly, I did like hundreds of eyes every year. So as soon as I open the eye, I can feel it. It's a good quality or not good. It's the retina is not good at all. So
Okay.
that's how we decided
Just
it.
have Yeah, I just have one more question. Do you actually oxygenate the explant prior to plating them?
No, actually,
Mhm.
I don't think it's necessary because we have a very thin layer of the median on top of it. So I think and it's open air region, so I think the oxygen level should be enough. Of course, the best way is we still try to test it. We never did this part before, so it is under our like plans to try to get oxygen detectors to see how the oxygen can penetrate these perfusion systems.
Thank you.
Hi, Prof Ling. I'm Hui Wen from I'm part of the Retina team. Congratulations on the amazing work that your team is doing. I have a couple of questions. So first is from the earlier part of your talk, you alluded to the use of lipid nanoparticle in delivery of I can't recall what, I'm assuming drug delivery into the photoreceptor. Is there a specific type of lipid nanoparticle that you're using?
Yes. So basically, we tried with our collaborators, we tried around 20 different formulation of the lipid nanoparticles. So we find two formulation. One is more preference to get the photoreceptors, while the other is more preference to get into the Müller cell. But I want to clarify here is the data I showed actually is in vivo data in mice. When we try to on the X plant, actually we found the X plant is a different story. So we believe the different formulation of the lipid nanoparticle actually will behave quite significant difference in vivo, ex vivo, and also
Sure.
in mice or in human.
Okay. And then, well, pertaining to MacTel specifically, so you showed that the expression of the deoxysphingolipid as well as the alanine levels were highly expressed in the nuclear layer. And we know that in macular telangiectasia, although it's a neurodegenerative disease, that we also get photoreceptor cell death. And but these the levels of these chemicals, were they also highly expressed in the photoreceptor? Because I don't recall seeing in the slides that you.
Okay, so this is a very good question. So this is still we are not very clear. One thing we are quite confident is it's a primary it's Müller cell the primary disease. So Müller cell dysfunction is primary disease. So Müller cell will be the first one who has dysfunctions, then eventually cause the photoreceptor death. While we can see that no matter the SPT, the key enzyme of the palmitoyl CoA react with serine or alanine, and the ALT and all the other PHGDH, which is the key enzyme for the serine metabolisms, these enzymes are all located in the Müller cells. So it seems like Müller cell plays a key role to control all these metabolisms. While the deoxysphingolipid experiment, maybe I didn't really present very clearly. We just test with adding the exogenous deoxysphingolines. So that causes the toxicities. It's not the endogenous ones. We still don't know the endogenous ones where it could accumulate because the result we got is the whole retina like a bulk lipidomics. It's not the single cell or spatial one. But the exogenous ones already show the toxicity to the Müller cell, and it's contradicts to most of the prediction because Müller cells as a glial cell should be very resistant to different kind of stress. While we don't know why this selected killing Müller cells, it could be maybe for some other things. While it still need further study on this part.
And if I may, two more questions. In
No
the conversion from pyruvate to alanine and by suppressing that, do you think there's a dose dependent? Like the amount that is because you see that in your graph, you do see a clear drop in the level of alanine, but I just wonder whether it's dose dependent and if it's something that can be completely suppressed, and therefore if it is, is that something that we worry about?
Actually, yes, we at the very beginning, that's why we do the safety test first because we believe, okay, if the macula need alanine, large amount of alanine, it must be the reason there. And if we inhibit the alanine, like generate alanine, it could be potential a big problem. The experiment here, there's a kind of disadvantage is because people until now the field know very little about the real concentration of the alanine, real concentration of the serines inside the retina. How like this is what we are studying actually right now is how the nutrition can pass the RPE and fueling the new retina or the new reverse back some nutrition back to the RPE. So all this data is actually coming out from like ex vivo culture conditions, I cannot say it's really happening inside the in vivo or not. Well, but the biochemistry level, it's very clear. So if we block this, the new retina has some response, and the blocking the alanine synthesis actually seems do not have very significant changes of most of the function of the new retina. I didn't show the one of the negative data here is we also do the RNA sequence profiling after the BCL treatment, whether there's any gene profiles expression changes. Well, very little genes changes. So it seems at least temporary inhibitions is very safe and stable. Well, the curve I didn't try. I think it's a good suggestions. We should try some different dosage to see which one is more efficient.
Okay, and one last question, if I may, and this pertains to the trial in giving patients serine. It's oral serine, right? In the
Yes.
clinical trial.
Yeah, they are doing.
And so, in the absence of the knowledge of how much of these serine and alanine that we need, at least in the eye, how much is actually delivered, how much ocular penetration we will get, and therefore then how much impact do we think we will see? I suppose there is no harm in feeding up, in administering the serine, and whether excess amount of serine will have any side effects that we are not aware of. So I just wonder whether, because of the lack of knowledge in terms of levels that we require,
I don't know.
the
I don't
impact
know
that
this
we
answer.
intend to see.
I don't know the answer for the real change, the macula, because it's really hard. They do know, and also there's still one argument is the de novo serine sometimes play another very important role, not only the serine level. So they try to provide the exogenous serine, which will play very important role to change the deoxysphingolipid levels. While how the response for the de novo serine, so endogenous serine synthesized changes corresponding to this extra serine supplements, we also don't know. So this is still a lot of things we need to study.
Yeah.
Thank
Okay.
you so much. Congratulations.
Thank you.
Thank you. Thank you, Prof. Ling. It's very nice. So thank you also for the panelists. So before I close the sessions, I have two more things to remind. So everyone, you can rewatch the webinars from our YouTube channels, and we need more followers. And second thing is there will be another webinars in this month, 24th of February, at the same time. We are inviting Prof. Alison McGrath. She will talk about the novel ways to investigate functional loss in the glaucomas. Okay, thank you very much again to being here. Have a great night. Yeah, so see you next time.